Hello and welcome to this high-level introduction to the NIH Guidelines and the Institutional Biosafety Committee. This presentation will provide you with an overview of the NIH Guidelines document, the role and responsibility of the IBC and its members, and discuss the specific information you should have regarding your organization's IBC. The goal of this presentation is to help you better understand your role on the IBC. What a risk assessment for an IBC protocol should include, resources to help you perform your review, and the types of experiments covered under the NIH guidelines.
Sometimes it's important to know where you've come from, to understand where you are. So let's begin. The NIH Guidelines on Research Involving Synthetic or Recombinant Nucleic Acids, often referred to as just the NIH Guidelines, contains recommendations from a number of federal government entities. Although they are called guidelines, the recommendations contained in the document become regulation under certain circumstances. These will be described later. This document details the safe and responsible handling of genetically modified materials. As mentioned, the guidelines source their guidance from the rules and recommendations from a number of organizations. These include the National Institutes of Health, Office of Scientific Policy, the Department of Health and Human Services, the Public Health Service, and the Occupational Health and Safety Administration, or OSHA.
Before we get too deep into all of that, let's talk about why the NIH developed these guidelines. Concern over this kind of research began back in the early 70s, when genetic engineering was gaining momentum in the scientific research community. In 1974, there was a biochemist at Stanford named Paul Berg, who later went on to win a Nobel Prize for chemistry. In an experiment, Dr. Berg was able to combine DNA from two different viruses, a monkey virus called SV40, and a bacterial virus known as Lambda. In his final step, he was going to place this new, genetically modified or recombined virus into a laboratory strain of the bacteria, E. coli. Due to the pleas from fellow investigators, he did not complete this last step. This work and the new potential biohazard resulted in an international conference, held in 1975, to discuss the potential biohazards and risks involved with this new recombinant technology. The guidance that eventually came out of this conference was a set of guidelines, now known as the NIH Guidelines for Research Involving Recombinant or Synthetic Nucleic Acid Molecules.
It should be noted that this historic meeting was called by concerned, engaged scientists. Not politicians, not religious groups, not the press. Concerned scientists were looking to have other concerned scientists review and regulate the research. The guidance that eventually came out of this conference was a set of guidelines that have become the NIH guidelines we are talking about today. It is from this document that we are charged with establishing an Institutional Biosafety Committee, or IBC.
Let's take a quick detour to talk about the other major institutional committees you may have heard of, so you can better understand how the IBC fits into the bigger picture. The three main committees involved in research oversight are the Institutional Review Board, or IRB, the Institutional Animal Care and Use Committee, or IACUC, and the Institutional Biosafety Committee, or IBC. While these committees may occasionally review the same research protocols, their core focus areas, governing regulations and operating procedures differ. The IRB is focused on ethics and the protection of human subjects, regulated primarily by Health and Human Services and the FDA under the Common Rule, 45 CFR 46. The IACUC ensures animal welfare in research, following USDA animal welfare regulations and PHS policy. While the IBC is concerned with protecting researchers, the community, and the environment when work involves genetically modified biohazards, guided by the NIH guidelines. Each committee operates under different federal mandates, and procedures allowed in one may not be permitted in another. For example, expedited review, which is allowed under IRB rules for minimal risk research, is not permitted under NIH guidelines for the IBC. The NIH guidelines document is what we call a living document. As science and technology evolves, the guidelines are revised and updated to reflect our current knowledge. The last update was in April 2024. The guidelines document is fairly lengthy, 132 pages at the time of this writing, and defines much more than just what constitutes recombinant and synthetic nucleic acids and who makes up an IBC. The guidelines document explicitly describes the different levels of protection or containment for worker safety and protection of the environment. This includes the safety procedures, practices, and facilities required for handling these recombinant materials. Furthermore, the NIH describes the roles and responsibilities of each party involved in the research and IBC review, which we will cover later in the training. And all of this are just the first 4 sections of the document. In addition to the 4 main sections, there are 13 appendices that provide additional guidance.
No need to panic. We aren't going to go over every section. We will address only the sections that are relevant to your role as an IBC member. So what information in the guidelines is relevant to your role as a committee member? Well, let's start with the committee itself, outlined in Section 4. The guidelines specify that any institution that meet the criteria set forth in Section 1 must establish an Institutional Biosafety Committee. That's you. The rules for the committee are: the IBC must have at least 5 members who have adequate experience and training to provide a comprehensive review of the research. 2 of these members are chosen to be the voice of the community. They must be local, within 50 miles, and completely unaffiliated with the institution. This excludes relatives of faculty and staff and former employees. Experts in specific pathogens or techniques, animals, plants, human clinical trials, or other specialized research areas must be available to review relevant research. Whether this means as full-time members of the committee or experts brought in on an as-needed basis. The IBC is charged with the review and approval of applicable research protocols, continued oversight of approved protocols, as well as ensuring researcher compliance with the guidelines and reporting any accidents, exposures, or breaches of containment to the NIH OSP. Your IBC chair, IBC coordinator, or biosafety officer will guide you in any potential noncompliance or reporting matters, and it's a great topic for continuing education. So let's talk about protocol review, since this is the work you'll do independently.
The IBC is directed to review all research projects that fall under Section 3 of the guidelines. This review includes an independent assessment of the biological agent, the genetic modifications, the facilities, procedures, training, and expertise of personnel involved in. For clinical research, an assessment focused on biosafety issues. Safety, not human subject ethics considerations. It is through an assessment of all of these elements that the committee will determine the containment practices and biosafety levels required. The guidelines provide a fuzzy roadmap of how to perform this review. I say fuzzy because there are many portions of the guidelines that are up for interpretation. This is by design, to allow organizations the flexibility they need to work the guidelines into their resources. Let's explore Section 1. What do the guidelines cover anyway?
The guidelines define recombinant nucleic acids as molecules that are constructed by joining nucleic acid molecules that can replicate in a living cell, like our earlier example with Dr. Berg's experiment. Synthetic nucleic acid molecules include those that are chemically synthesized or amplified, as well as those that are chemically or otherwise modified, but still capable of base pairing with naturally occurring nucleic acids. This category also includes molecules that result from the replication of any of the above. Conducting this type of research alone does not automatically require IBC review. The source of funding is also a determining factor. For now, it is important to understand that if you are taking this training, any research at your institution that meets these criteria falls under the purview of the IBC. With that foundation in place, let's look at how the review process itself is performed.
Section 2 of the guidelines explains that the IBC must perform a comprehensive risk assessment of the research. This means looking at several factors: the parent organism, the proposed genetic modifications, experimental procedures and equipment, waste handling, and the facility where the research will be conducted. Using this information, the IBC determines the best ways to protect the researcher, the community, and the environment. This is where the appendices come in. They provide a roadmap for performing the protocol review. Appendix B lists the risk group for each agent. Appendix G describes the standardized biosafety containment levels. And Appendix I reviews biological containment. Moving on to more specialized research, Appendix L discusses plant work while Appendix M talks about animal research. and Appendix K applies to large-scale work.
There is so much to know about performing a comprehensive risk assessment and employing biosafety containment principles and practices that we aren't going to cover it in this training. The NIH mandates not only NIH Guidelines training for IBC members, but also periodic refresher training. So, we are going to address risk assessment and some other meatier topics in upcoming training sessions. Until that time, and even after you have undergone further training, as you review protocols, utilize the resources you have at your disposal. These include the appendixes from the guidelines, the CDC's publication, Biosafety in Biomedical and Microbiology Laboratories, or the BMBL. And there are great international resources, such as the World Health Organization's Laboratory Biosafety Manual or the Public Health Agency of Canada's Pathogen Safety Data Sheets. Don't forget to reach out to your biosafety officer, and of course, each other. The reason the IBC is a committee and not one person is to allow for discussion of biosafety principles and practices for each research project.
There is one last part of the NIH guidelines we need to cover today, and it's an important one. Section 3, Experiments covered by the guidelines. This section matters because it is the IBC's responsibility to verify how a research project is categorized and to document that decision in the meeting minutes. Section 3 is divided into 6 parts, labeled A through F. Now, you'll notice we are not going to review sections 3A or 3B in this training. This is because these sections address experiments that are reviewed directly at the federal level and are not typical of the research reviewed by most IBCs. For that reason, most IBC members will not encounter them in their day-to-day work. Instead, we will begin with Section 3C, which is the first category typically relevant to IBC review, and even then, not all IBCs encounter 3C experiments. Section 3C are human clinical trials that involve the deliberate introduction of recombinant or synthetic nucleic acids into a person. According to the latest landscape report on cell and gene therapies from the Association for Cell and Gene Therapy, there are over 4,000 investigational genetically modified therapies, somewhere along the translational medicine pathway. Clinical research with these products falls under Section 3C of the guidelines. Sections 3D, 3E, and 3F are distinguished by 2 things: how and when the principal investigator must notify the IBC, and whether approval must be granted before the research begins. It's important to note that at some institutions, all research must receive IBC approval before work can begin, regardless of its NIH category. This provides consistency and ensures nothing slips through the cracks. You will need to review your charter or check with your IBC administrator to be sure. As mentioned previously, the NIH Guidelines is a living document. They are updated regularly, and there are many details to consider when determining the correct category for a protocol. That's why it is essential for the IBC to confirm the research category identified by the PI and to document that determination clearly in the meeting minutes. Section 3D describes experiments that require IBC approval before any work begins. This category covers many basic microbiological techniques involving risk group 2, 3, or 4 agents, as well as restricted agents. These experiments usually require BSL-2 containment or higher. Most of the research in IBC we'll review falls under this section. Let's look at 3 common types. 3D. 1 are experiments that use RG2 or higher agents as hosts and involve introducing recombinant or synthetic nucleic acids into pathogenic organisms. 3D2 experiments where DNA from RG2 or higher agents is cloned into non-pathogenic organisms such as bacteria, yeast, or lower eukaryotes. 3D3 infectious or defective viruses in tissue culture. Experiments that use viral vector systems where a replication-competent virus could be reconstituted. This section also includes genetic modifications of whole animals or plants, and experiments using genetically modified agents in animals or plants, human influenza viruses, large-scale research and gene drive experiments.
Experiments in Section 3E are those that require IBC notice when work begins. Again, it is important to note some IBCs require approval before work may begin for any experiment, regardless of the classification. This section is somewhat of a catch-all for experiments that do not fall into the 3D category and are not exempt. Using a risk group 1 AV vector without a helper virus, or cloning GFB into certain bacterial strains. Yes, these are 3 experiments. It is important to mention that some simple, standard, molecular biology procedures might require IBC review. Some strains of E. coli, like K12, are exempt from the guidelines, while others, like BL21 strains, are not and require submission of an IBC protocol. Section 3F of the NIH Guidelines describes categories of experiments that are exempt from IBC oversight. How exempt experiments are handled varies by institution. Some organizations use a decision tree that allows researchers to determine exemption. Other institutions require all exempt experiments to be submitted for review by the Biosafety Officer, or IBC. Many fall somewhere in between. Examples of exempt experiments include nucleic acids that cannot replicate, modify genomes, or produce harmful toxins. Nucleic acids not associated with organisms or delivery vehicles capable of entering cells. Sequences that come entirely from a single natural source, or from hosts that naturally exchange DNA, such as certain prokaryotic or eukaryotic systems. Genomic DNA with non-recombinant transposable elements. One important exception: any synthetic nucleic acid transferred into a human participant may not qualify for exemption if it meets criteria outlined in Section 3C.
As we wrap up, it's important to acknowledge that there is much more to the NIH Guidelines and IBC protocol review than what we've covered today. This training was designed as an introduction to help you understand the guidelines at a high level, the role of the IBC, and your responsibilities as a committee member. In our short time together, we've focused on the most important sections you'll encounter as you serve your organization and your community. We strongly encourage you and your committee to take advantage of the NIH requirement for refresher training to explore other relevant areas in greater depth. There is also far more to learn about risk assessment, risk mitigation principles, and emerging technologies in biosafety. Those topics and more will be addressed in additional Azealia Labs training sessions. As you move forward, remember that every IBC is unique. Be sure you know. Scope of review. Does your committee review all biological research, only RG2+ agents, or just recombinant/synthetic materials? Institutional practices. How are FOIA requests handled? What is the review and approval schedule? Who communicates with PIs? Special considerations. Does your IBC review large-scale work, high containment research, or clinical protocols requiring specialized expertise? Governance and best practices. Has your IBC recently reviewed its charter? Are you aligning with the latest NIH guidelines and incorporating evolving best practices? The NIH guidelines establish many requirements but also allow flexibility. Each IBC has the ability and responsibility to structure its oversight in a way that ensures both compliance and efficiency for its institution. Thank you for joining today's training. As an IBC member, you play an essential role in ensuring research is conducted safely and responsibly, protecting not just the researchers, but also the community and the environment.
If you have questions, please reach out to your IBC staff for more information about your committee, the NIH guidelines, or your role in oversight.
We also encourage you to continue building your knowledge through additional training. Azealia Labs offers ongoing sessions as well as custom, personalized training modules designed to fit the needs of your committee and your institution. We look forward to seeing you in future trainings and to supporting you as you grow in your role as an IBC member.